वेलकम टू केपी पॉइंट्स मित्रों। केपी पॉइंट्स में आपका स्वागत है और यहाँ पे हम डिस्कस करेंगे प्लांट फिजियोलॉजी एंड बायोकेमिस्ट्री का लेक्चर 1 जो कि है बीएससी और एमएससी स्टूडेंट्स के लिए। ठीक है मित्रों, चलिए जिसमें क्वेश्चंस यूनिट 1 से लेते हैं। मेम्ब्रेन ट्रांसपोर्ट, मिनरल न्यूट्रिशन एंड एंजाइम्स। फर्स्ट क्वेश्चन, ट्रांसपोर्ट ऑफ सुक्रोज into the plant vacuoles against its concentration gradient is typically driven by which mechanism? So this is antipode with protons H plus moving down their gradients into the cytosol. Question number 2, underwater deficit stress guard cells loss turgor. The initial step triggering this turgor loss involves the opening of which channel? Inward rectifying potassium channels? No. Anion channels SLAC1 causing depolarization and subsequent potassium efflux? Yes, correct answer is B. Now question number 3, if an enzyme follows Michaelis-Menten kinetics, जिसे हम Km constant बोलते हैं कि नहीं बोलते, what happens to the apparent Km and Vmax in the presence of an uncompetitive inhibitor? So answer is Km increase and Vmax remains unchanged. No. Both Km and Vmax decrease proportionally. Yes. Question number 4, the active site of enzyme undergoes structural refinement upon binding to the substrate. This behavior is best described by which model? So this described by lock and key model. No. Induced fit hypothesis. Clear? Question number 5, plant nitrate reductase is a homodimeric enzyme. Which combination of prosthetic groups / coagents is essential for its catalytic activity? So answer is FAD, heme and molybdenum cofactor MoCo. Question number 6, when a reaction rate is measured at a substrate concentration equal to Km by 4, the initial velocity will be, so initial velocity will be. Number 7, which of the following is an example of an allosteric enzyme regulatory mechanism commonly observed in plant amino acid photosynthesis pathways? So answer is competitive binding of structural analogs at the active site. No. Feedback inhibition where the final pathway product binds to the distinct regulatory site. Yes. Now question number 8, the entry of boric acid H3BO3 into plant root cell across the lipid bilayer occurs predominantly via primary active transport pumps? No. Group translocation? No. Channel mediated facilitated diffusion or passive modulin 26 like intrinsic? Yes. So correct answer is C. Now question number 9, which of the following student statements regarding its isoenzymes or isozymes is correct? So which one is correct? They possess identical primary amino acid sequences but catalyze different reactions? No. They are different oligomeric forms of completely unrelated proteins acting on the same substrate? No. They catalyze the same chemical reaction but differ in kinetic parameters Km and Vmax and electrophoretic mobility. So answer is C. Now answer question number 10, the spatial distribution of water potential psi W across plant tissue indicate the water will always move exponentially towards region with more positive solute potential psi S? No. High less negative total water potential? No. Lower more negative total water potential. So correct answer is C in case of 10. Now question number 11, the operational mechanism of p-type H plus ATPase in the plant plasma membrane involves the transient formation of A, phosphorylated aspartyl residue intermediate. Question number 12, the zinc deficiency symptom known as little leaf. Important है. The zinc deficiency symptom known as little leaf or rosette habit in plants occur primarily. Zinc is a structural cofactor for the synthesis of, so chlorophyll B? No. Tryptophan, the direct biochemical precursor of indole 3-acetic acid oxygen? Yes, answer is B. Now question number 13, in a classic Lineweaver-Burk double reciprocal plot, the value represented by the intercept on the horizontal axis is -1 by Kn. Yes. So correct answer is B. Question number 14. The translocation of silicon Si in plants, which provides a mechanical strength and biotic resistance, is heavily concentrated in the cell walls of phloem sieve tube animals, no. Cortical parenchyma cells nuclei, no. Epidermal cells and trichomes as phytoliths. Yes, so correct answer is C. Now question number 15. Aquaporins can be gated, closed or opened post-translationally in response to environmental stimuli like flooding. This gating is typically regulated by phosphorylation of specific serine residue and pH drops in the cytosol. Question number 16. Ribozymes differ from conventional protein enzymes because they do not possess a specific tertiary structure, no. Are composed entirely catalytic RNA molecules. Important question. Question number 16. Question number 17. The mechanism of stomatal closure induced by elevated internal CO2 concentration depends on sudden activation of inward rectifying potassium channels, no. Massive hyperpolarization, no. Depolarization triggered by calcium dependent activation of outward anion fluxes. Yes, so question number 17, answer is C. Question number 18. Molybdenum is absolute nutritional requirement for plants because it directly constitutes the reactive core of cytochrome, no. Nitrogenase and nitrate reductase. So answer is B. Question number 19. An enzyme reaction exhibiting a Hill coefficient NH greater than 1 suggests hyperbolic kinetics, no. Positive cooperatively where substrate binding enhances the binding of subsequent substrate molecules. Answer B. Now question number 20. Solute transport via secondary active transport system utilize energy derived from. So energy derived from the dissipation of a pre-established electrochemical portion gradient. Answer is C. Now questions unit 2nd, photosynthesis and carbon assimilation. Question number 21. The non-photochemical quenching MPQ mechanism protect the photosynthetic apparatus from the excess light energy by dissipating it as heat. This process heavily relies on the operational conversion of xanthine to zeaxanthin via xanthophyll cycle. Answer is A. Question number 22. During photosynthetic electron transport, the reduction of plastocyanin PC is directly carried out by photosystem second, no. The cytochrome B6F complex via the Q cycle mechanism. Question number 23. In C4 plants, utilizing the NAD-malic enzymes NAD-ME pathway for carbon assimilation, the decarboxylation of malate takes place in which specific compartment? So takes place in bundle sheath mitochondria. Question number 24. The oxygenase activity of Rubisco increased preferentially over its carboxylase activity under which environmental conditions? In general, carboxylation activities are more greater than oxygenase activity. So answer is low CO2 availability, high O2 levels and elevated temperatures promote Rubisco oxygenase activity. So oxygenase activity की जो condition है वो क्या है? Oxygen ज़्यादा हो, CO2 कम हो. Now question number 25. The synthesis of starch inside the chloroplast stroma is catalyzed by ADP glucose pyrophosphorylase. The enzyme is allosterically activated by inorganic phosphate, no. 3-phosphoglycerate means 3-PGA. Question number 26. Which of the following photosynthetic pigments lacks a phytol tail and act as a water-soluble light harvesting accessory complex in cyanobacteria and red algae? So answer is phycobilins. Example, phycoerythrin. Question number 27. The cyclic electron flow around photosystem PS1 generate protons gradient across the thylakoid membrane without synthesizing NADPH. The electrons are routed from ferredoxin back to the plastoquinone pool via. So This is the PGR5, PGRL1 complex or chloroplast NADH complex. Answer is A. Question number 28. During the Calvin cycle, the regeneration of one molecule of ribose 1,5-bisphosphate RUBP from triose phosphates directly requires the consumption of one molecule of ATP in case of regeneration. Question number 29. Crassulacean acid metabolism CAM or CAM plants exhibit a diurnal rhythm in titratable acidity. Maximum level of vacuolar acidity is typically reached at the end of the day period, no, exactly midday, at the end of dark period, early morning. Answer is C. Now question number 30. The primary electron donor that reduces the photo-oxidized P700 plus reaction center of photosystem 1 is reduced plastoquinone, no, plastocyanin PC, yes, correct answer is B. Now question number 31. The structural organization of the light harvesting complex LHC second allows it to undergo state transition when LHC light harvesting complex second is phosphorylated due to over reduction of the plastoquinone pool PQ. It is physically migrated from the grain stalks to the non-appressed stroma lamellae to the associated with PS1. Question number 32. The carbon assimilation pathway of C4 plants effectively eliminates photorespiration by mechanically concentrating CO2 around rubisco in bundle sheath cells to saturate the carboxylase activity. So answer is B in case of 32. Now question number 33. The enzyme that converts glyceraldehyde 3-phosphate into ribulose 5-phosphate via structural reorganizations during the Calvin cycle includes. So answer is transtetolase and aldolase. Answer is A. Question number 34. The photolysis of water by the oxygen evolving complex OEC releases protons H plus. These protons accumulate directly inside which compartments? Chloroplast, stroma, no. Matrix, no. Thylakoid lumen, yes. Answer is C. Question number 35. The synthesis of sucrose in the plant cytosol is strictly regulated by the regulatory metabolite. So answer is ADP glucose, no. Fructose 2,6-bisphosphate. Answer is fructose 2,6-bisphosphate. 36. The structural arrangement of leaves showing distinct mesophyll cells encircling tight, large bundle sheath cells containing prominent chloroplasts is known as. So answer is Crange anatomy found in C4 plants. Question number 37. Adsorption of light by the reaction center P680 leads to the immediate ejection of an electron. The primary electron acceptor is structurally localized within the PS second reaction center. Core is. So answer is plastoquinone, no. Pheophytin, a chlorophyll lacking magnesium. Answer is B. Now question number 38. In C3 plants, the theoretical maximum quantum yield of photosynthesis drops as leaf temperature increases because thylakoid membranes become completely impermeable. The solubility of CO2 decreases, yes, faster than the solubility of O2, promoting photorespiration. Question number 39. The reduction of one molecule of NADP plus to NADPH during non-cyclic photophosphorylation requiring the sequential translocation of how many electrons through the Z scheme? Answer is 4 electrons through the Z schemes. Question number 40. The light-induced activation of several Calvin cycle enzyme is mediated by the thioredoxin system. Thioredoxin sense the reduction state of the electron transport chain by accepting electrons from. So NADPH, no. Reduced ferrodoxin, yes. Now questions from unit 3rd, respiration, lipid, nitrogen and secondary metabolites. Question number 41. The alternative oxidase AOX pathways in plant mitochondria transfer electrons directly from ubiquinol to oxygen. Ubiquinol to oxygen. The major physiological outcome of pathway is maximizing the synthesis of ATP per molecule of glucose oxidized, no. releasing energy as heat, thermogenesis, and preventing the over-reduction of the ETC to suppress ROS production. So answer is B. Question number 42. During the conversion of fat reserves to carbohydrate in germinating oil seeds, succinate exported from the glyoxysomes enters the mitochondrion to be converted into. So answer is oxaloacetate. Now question number 43. The assembly of the core structural backbone of fatty acid within plants, plastids, is carried out by fatty acid synthase. In plants, this enzyme complex is structurally organized as large multifunctional no multi-enzyme complex where each reaction is catalyzed by a distinct separate protein. Answer is B. Question number 44. The reduction of nitrate NO3 minus to nitrite NO2 minus occur in the cytosol, while subsequent reduction of nitrate to ammonia NH4 plus occurs inside the plastids / chloroplasts. Yes and no? Yes. Question number 46. The structural precursor for the biosynthesis of all phenolic compounds and flavonoids by the phenylpropanoid pathway is higher plants in the amino acid. Tryptophan, no. Phenylalanine or tyrosine in some plants, yes. Answer is B. Question number 47. The primary pathway responsible for providing erythroid 4-phosphate, a key structural intermediate needed for the biosynthesis of aromatic secondary metabolites via the schemate pathway is. So answer is glycolysis, no. The pentose phosphate pathways, B. 48. The biochemical steps of plant mitochondrial beta oxidation differ structurally from the glyoxymal pathway because mitochondrial acyl coenzyme dehydrogenase passes electrons directly to oxygen produce hydrogen, the respiratory electron transport chain via FAD, converging energy as ATP. Answer is B. Question number 49. The nodulation factors, node factors, secreted by symbiotic rhizobium bacteria to initiate nodule formation in legume roots are structurally classified as. So structurally classified as small lipochitin oligosaccharides. Question number 50 and last of this lecture. The synthesis of isopentenyl diphosphate IPP, the universal 5-carbon building blocks, all terpenoids via the plastidial MEP pathway originated from 2 molecules of acetyl coenzyme A, no. Pyruvate and glyceraldehyde 3-phosphate. Answer is B. So part 1 of top 100 question plant physiology and biochemistry is complete, important for B.Sc and M.Sc students. And next part 2 from 51 to 100. Clear?